right, we're here today to talk all about meiosis, which is going to be the second type of division that we discuss. So just a little bit of a reminder on some of the vocab that we've talked about. Mitosis produces two diploid somatic cells. Now that means in the end, it takes one cell and makes two. They are diploid, meaning they have the full set of genetic information, 2 copies of each chromosome. And they are somatic cells, meaning they are body cells. And these cells are going to have the same genetic information as each other. They're going to have the same number of chromosomes as each other. And as the parent cell, meaning we are making identical copies. So I like to say MI, I in mitosis, stands for identical copies. Therefore, because we're making identical copies, mitosis is a type of asexual reproduction. And the other type of asexual reproduction we talked about was binary fission and prokaryotes. But binary fission was much more simple than mitosis. So again, the last type of cell division we talked about was mitosis. And there we're going to make 2 identical cells. Those 2 identical cells are going to be diploid, meaning they have the exact same number of chromosomes. They have 2 copies of each, the normal number of chromosomes, which is 2 copies of each chromosome. And the cells that they are creating are somatic cells, which are body cells. And remember, body cells are going to be cells like in the arm, stomach, pancreas, liver, brain, everywhere in your body except your sex cells. So mitosis creates 2 diploid somatic cells, and they're identical copies. Therefore, this is a type of asexual reproduction. So just in a background on sexual versus asexual reproduction. Asexual reproduction is very primitive. It's simple. It was the first type of reproduction ever. Whereas sexual reproduction requires a complex series of cell divisions. And we're going to talk about that today. Asexual reproduction can make large numbers of offspring without a partner. Asexual reproduction can be done individually. There is no need for a partner. Whereas sexual reproduction requires more time and a partner. Asexual reproduction leads to 0 genetic diversity because it's essentially creating clones. If you're making clones, there's no diversity. In sexual reproduction, there is lots of genetic diversity. So that's a background. Asexual would be mitosis, which we already talked about. And sexual is going to be what we talk about today, which is meiosis. And the way that you can remember that is the E in meiosis can easily be changed to sexual reproduction. So the E in there stands for sexual reproduction. Now you might be asking, well, we already learned about one type of division. Why can that type of division not be used to make gametes? So why can we not use mitosis to make sperm and egg? And the reason for that is actually kind of simple when you think about it. We know that humans have 46 chromosomes. When we look at humans, they have 46 chromosomes. And that's normal. That's the normal number. If you're human, you should have 46. Now, if we used mitosis to make our sperm and our egg, The sperm would have 46 chromosomes. The egg would have 46 chromosomes. When the egg and the sperm come together to make the baby would have 92. That is not correct. If a baby had 92 chromosomes, well, they would not even exist. That is not possible. That does not happen. This is not going to be the process to make humans. Therefore, mitosis cannot be used because if it was mitosis, the baby would have the wrong number of chromosomes. And that's going to lead us to the process of meiosis. So meiosis is a special type of division that occurs only in sexually reproducing organisms. So it happens only in sexually reproducing organisms, and it takes the normal number of chromosomes and reduces it by half. So it would take a cell with 46 and make a cell with 23. So it reduces the number in half. Therefore, meiosis makes haploid. Remember, haploid means a cell with half the number of chromosomes, HA for half, and it makes gametes. And remember, gametes are sex cells. So it makes sperm in males that have 23 chromosomes, and it makes eggs. You might also see the word ova, and females that again also have 23 chromosomes. So when you take human normal number is 46, we go through the process of meiosis. We have a sperm with 23. We have an egg with 23. We add them up and we get 46 in the baby. And that is our magical number that works. Therefore, meiosis is going to be used to make sperm and egg. It's used to make sperm and egg, so they have half the normal number of genetic material, half the number of chromosomes. That way, when you add the sperm and the egg and you mix them together, the baby will have the normal number. All right, so first we're just going to go over a simple overview of meiosis. Meiosis involves 2 rounds of division. The first round of division separates homologous pairs. And the second round of division separates sister chromatids. Now, the reason we have 2 rounds of division is because the process is similar to that of mitosis. So we start with 46. First round, we make copies. The 2 cells in the end have 46. But again, that is not half yet. So we have to do it to make 23. So we go around 2 rounds of division so that in the end our cells are fully haploid. And we'll talk about what that means, fully haploid, here in just a second. So the steps of meiosis are going to look similar to those of mitosis. They have the same names. The only difference is there are 2 rounds, and each round has a number associated with it. So if you see the name of a stage and it says prophase 1, if it has a number, you know it's meiosis, because mitosis does not have numbers associated with it. So preparing for meiosis to happen. Before meiosis can start, DNA must be replicated. So we have to make a copy of the DNA before meiosis, and that's going to happen in S phase of interphase. So this is exactly the same as that of mitosis. DNA has to be copied first. After the DNA is copied, the cell will move into the first prophase. So it will move into prophase 1. During prophase 1, homologous chromosomes come together to form what is known as a tetrad. When they're in a tetrad is essentially 4 chromatids all smushed together like we see here. They come very, very close to one another. And we have these color-coded, let's say blue from dad, red from mom. They come very, 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 very close to one another. So close that the chromosomes can exchange parts. So they come so close that the chromosomes exchange pieces of each other. And what this does is it creates new combinations of traits. And this is one of the things that leads to genetic diversity. So when the chromosomes come together in prophase 1, they get so close to each other that bits and pieces can mix and match. And that mix and matches traits in offspring. And that is why parents don't have all identical children, because their sperm and their egg are different. Because of crossing over, each sperm and each egg are unique, even if they come from the same person. So a dad is not going to have all of his children look the same because all of his gametes are different due to crossing over. So that leads to what is genetic diversity. So this part here, the exchanging of homologous chromosomes, that is called crossing over. And crossing over is going to be one of the keys that lead to genetic diversity in offspring and in sexual reproduction. So after prophase 1, the cell moves to metaphase 1, which is the first metaphase. In this stage, the pairs of homologous chromosomes line up across the middle of the cell. Remember, M for meta means middle. In metaphase 1, we're going to see full duplicated chromosomes lining up in the middle. So we have pairs of chromosomes lining up in the middle, tetrads lining up in the middle. Then anaphase 1. Anaphase A still equals apart. In this case, though, we have full duplicated chromosomes pulling apart. So homologous chromosomes remember, these are chromosomes, not chromatids, separate. So there is a full duplicated chromosome here. Remember, A equals apart. Now we're going to move into the first telophase. In the first telophase, the chromosomes, again, full duplicated chromosomes, are moving to the poles. So they're at the edges of each cell. And cytokinesis will begin. Cytokinesis is the process of the cell splitting. Now, at the end of meiosis 1, we have 2 haploid cells with duplicated chromosomes. Now, you may be saying, Miss Ray, we needed haploid cells to begin with. They are haploid now. Why do we not stop? And the reason is because, yes, the cells are haploid, but each chromosome has an identical copy. So while the cell is haploid, there is an extra copy of every chromosome. Therefore, while the cell is haploid, there's still an extra copy of everything. We do not need those extra copies. Therefore, we're not done yet because we still need to split these duplicated pairs in half. Now, because of this, the cells do not replicate the DNA in between meiosis 1 and meiosis 2. If we were to copy the DNA again, we would just be right back where we started. We would not be on our way to making our haploid cells. We do not want to copy our DNA again. We only copy the DNA once at the very start, not in the middle. So after the cell splits in two, we immediately move into meiosis 2. Now, meiosis 2 is just like mitosis. They are going to look the same, exactly the same. We will have prophase 2, we'll have the spindle form. Metaphase 2, the chromosomes line up in the middle. Note, this time they are individual, they are not with a pair. Then the chromatids pull apart. Again, not the whole chromosome, but the individual chromatid. Then we move into telophase and cytokinesis. After cytokinesis, we have 4 unique haploid gametes. Each gamete is different from each other because of crossing over. There are 4 of them because we divided twice. If we divide twice, there's going to be 4 cells in the end. They are haploid, completely, fully haploid. And they are gametes, which means they are either sperm cells or egg cells. So we're going to go through a compare and contrast chart. So comparing and contrasting. Mitosis involves only one round of division. The cell splits once. Whereas meiosis has 2 rounds of division. When we are looking at identical cells, we are looking at MI, mitosis. Mitosis also makes 2 cells because they only divide once. Daughter cells are genetically unique in meiosis. Remember, unique in sexual reproduction. E stands for sexual here. Because we have 2 rounds of division in meiosis, it creates 4 cells. Haploid gametes are created in meiosis. Diploid cells are created in MI, mitosis. Somatic cells for growth and repair. Somatic cells is mitosis. Gametes or sperm and egg, again, that's sexual. I didn't mean to write sexual there, but it is ME. Remember, E stands for sexual reproduction. Crossing over gives genetic diversity. Therefore, that is meiosis. No crossing over is MI for identical copies, mitosis. All right, I know this is a lot and we're going to have some practice, but first I want to tell you about something cool that makes meiosis a bit different in males versus females. The process that we talked about just now is exactly the same, but each type of meiosis in males versus females, it has its own name. In men, it is called spermatogenesis because it's making sperm. In women, it's called oogenesis because it's making eggs in the ovaries. Both processes make 4 haploid gametes. The only difference is that the size of them is different. In females, oogenesis makes 1 large gamete, and the other 3 are small. But in spermatogenesis, to make sperm, all are the same size. So the only difference are the size of the gametes that are produced. Now we're going to just finalize with some of the sources for genetic diversity. So meiosis itself and sexual reproduction produce genetic variability or diversity. The first way that this happens is through independent assortment. Independent assortment happens during metaphase. So the homologous chromosomes can line up in any order. So if we look here, chromosomes can line up. They could have all the blue on one side, all the red on the other side. They could be mixed here. So in the bottom, these ones switched here. These ones are backwards. They can be in any order on any side. And the side that they line up on, that they assort into, will determine the traits that the gamete gets. So they assign themselves randomly. They split randomly. We don't know which side is going to get which variability from the chromosome. Another place that we get genetic diversity is crossing over. Remember, crossing over is when bits and pieces of the homologous chromosomes switch sides in prophase 1, and that's what makes all sperm and all egg unique. And then there's also the process of random fertilization, which means we don't know which egg and which sperm are going to come together. It's always random. So it's random fertilization. And these lead to diversity amongst organisms that reproduce sexually. All right, so that was a lot. There's going to be some practice on mitosis versus meiosis on Canvas, so go ahead and check that out.